Okay. Hi, I'm Michelle Graham. I'm an OMS too, and I did my research on podocyte cell death and if diabetes-associated triglyceride-rich lipoproteins contribute to that. And podocytes are basically these cells that line the glomerular capillary membrane and help with the filtration barrier. And in diabetic kidney disease, we know that it's associated with the loss of these cells and therefore leading to the loss of filtration. And as well as high lipids are associated with diabetes. So what we were trying to look at is if we could see the mechanism by which the podocytes are dying. So basically, our proposed mechanism was that the VLDL, which is a high lipid marker, that if that was being taken up by the podocytes and basically causing them to die. And we've seen in the lab that we do see the lipids in the glomeruli and inside the podocytes. So we thought, can we show that in the wet lab? So here's, I had BTBR mice, which are basically mice that were fed this high-fat meal, basically, and then they were induced human-like, dyslipidemia. And we had the diabetic mice and the non-diabetic mice. And we treated one of each group with this ApoC3 ASO, which is basically thought to lower the triglyceride amount. So to see if that would kind of show that that's protective in podocyte loss. Well, I took some images with immunohistochemistry of the glomeruli of these rats in all 4 groups, and I quantified the podocyte, which is a marker for podocytes, using ImageJ. And I also cultured the podocytes in vitro, so in the lab, and differentiated them to potentially see if we could load them with the VLDL. And we saw that diabetes results in podocyte loss. So those with diabetes had lower glomerular podocyte area and less podocyte per glomerulus. But we didn't see a statistically significant difference between those with the ApoC3 ASO, so that those that had that protective lowering of triglycerides. So that begged us to question, is podocyte loss mediated by a different mechanism? So, and we'd also like to continue exploring other mechanisms of podocyte death. And yeah, it was sponsored by the NIH, and I'm thankful for this opportunity.